This article introduces an innovative method for performing brain surgery on small animals, bypassing the need for a brain atlas or a stereotaxic frame. Traditional stereotaxic surgery, a cornerstone of brain research, relies on a standardized head position and a species-specific brain atlas to guide the placement of electrodes and other tools within the brain. However, this approach is limited to animal species for which these tools have been developed, thereby restricting research primarily to common laboratory animals. The resources required to develop a brain atlas and stereotaxic techniques for each animal model are extensive, making it difficult for many research labs to expand their studies to non-conventional animal models. This new technique offers an alternative by using a high-contrast magnetic resonance imaging (MRI) scan and a microcomputed tomography (CT) scan, coupled with a multi-camera tracking system. The core of the new method involves attaching miniature markers, or fiducial markers, to the skull of the animal. After the markers are attached, a CT scan of the animal's head is performed. Prior to this, an ex vivo high-resolution MRI scan is conducted on one animal of the species being studied. The MRI and CT images are then co-registered using standard image processing software. This process involves aligning the MRI image of the brain with the CT image of the skull, using the fiducial markers as reference points. The targets for brain recordings are marked on the MRI image. During surgery, the animal's head is stabilized in any convenient orientation, and the probe's three-dimensional position and angle are tracked using a multi-camera system. The system includes software that uses the on-skull markers to align the CT/MRI model with. This method has been successfully applied for neuropixels probe positioning in owls, quails, and mice, demonstrating its versatility. In more detail, the ex vivo MRI scan was performed on one adult female quail and one adult male barn owl, following a modified protocol. A 3D printed plastic frame with 4 cone-shaped holes and individual metal wires. When using a plastic frame, the cone-shaped holes are visible in the CT image and act as landmarks. For better contrast in the CT, metal wires can be inserted into the holes. With the other method, 4 small pieces of tungsten wire are embedded in dental cement at different locations on the skull, with care taken that they form a unique rigid body with 6 degrees of freedom and are not on a single plane. A fine drill is used to expose the upper tip of each marker. About a week after the first surgery, the animals undergo a CT scan after being anesthetized again. The head is fixed using X-ray inert foam and medical tape. CT scans are conducted using a micro-CT scanner for animal research at a resolution of 84 mvoxel. The three-dimensional volumes are reconstructed using software such as nRecon, MIPAV, and ImageJ. The next step is aligning the MRI and CT models. The MRI image has to be scaled and aligned to fit the CT scan using a process called volume registration. Open-source software packages such as Fujiyama or the MATLAB MRegform function are used. The alignment process is a 2-stage process involving manual and automatic registration. Manual registration allows for a rough superposition of the 2 datasets before automated algorithms find the optimal registration of the MRI image with the CT image. After registration, the coordinates of the fiducial markers are identified in the CT images and inserted into the MATLAB code. The brain targets are also identified in the registered MRI images and included in the code. The second surgical procedure involves positioning the electrodes in the brain at the correct location and angle. The animal is anesthetized, and the head is fixed, though the orientation of the head is not critical. Depending on the available hardware, either a mechanical XYZ positioning system or a 3-dimensional video-based tracking system can be used to carry out the surgery, or a combination of both can be used. When using a mechanical XYZ positioning system, a syringe needle is attached to the vertical arm of the manipulator to measure the coordinates of the fiducial markers. The measurements are fed into the computer, which calculates the estimated positions of the desired brain targets. The software provides the estimated XYZ position of the entry point on the skull and the desired angle of the probe. A craniotomy is performed to expose the brain surface, and the probe is inserted at the correct angle. When using a 3-dimensional video tracking system, a commercial motion tracker, OptiTrack, is used. The system consists of 3 to 4 infrared cameras, a laptop, and the motive software. A 3-dimensional printed measurement tool with 3 infrared reflectors and a needle at the tip is used to measure the coordinates of the fiducial markers. The MATLAB program uses the position of the infrared reflectors to compute the coordinates of the fiducial markers and then calculates a transformation to align the CT-based model with the surgical coordinate system. The program continuously reads the position of the reflectors and provides real-time feedback on the angle and position of the probe. A MATLAB program was developed to accompany the surgical procedure. Before the surgery, the coordinates of the fiducial markers in the CT, the brain targets, and the entry point need to be inserted in the code. The software displays a line connecting the two brain targets on the CT image, allowing the user to select the intersection point with the skull to mark the entry point. If optical tracking is not used, the coordinates of the fiducial markers are measured using the mechanical XYZ positioning system and entered manually into the program. If using optical tracking, the program streams data from the motive software and calculates the fiducial marker's XYZ coordinates. The program uses the coordinates to calculate the best transformation between the CT and surgery coordinate systems using an algorithm that applies singular value decomposition. It then displays a 3D plot of the transformed markers, brain targets, and entry point to assess the quality of the fit. Additionally, the program provides the coordinates of the entry point and two penetration angles. The program calculates and displays in real time the position of the probe tip and the angles of the probe. Histologically, NeuroPixel's probes are dipped in a dye solution before penetrating the brain to allow tracking of the probe after the experiment. After the experiments, animals are perfused with PBS and paraformaldehyde, then the brains are sliced, stained with DAPI, and imaged. To quantitatively assess the accuracy of the method, a simulation was performed, using noisy samples of fiducial markers. The simulation involved drawing marker positions from a normal distribution centered around the original marker positions, with a standard deviation of 100 m. This was done to mimic the error in positioning the markers using the optical tracking system. The algorithm was applied to calculate the transformation between the noisy sets of markers, and the transformation was applied to target points, and the estimated error was measured as the Euclidean distance between the transformed target and the original target. The simulation revealed that positioning accuracy decreases with distance from the frame's center, with a maximum expected error of 350 m located 5 mm to the side and 10 mm below the frame's center. Additionally, a physical assessment of the method's accuracy was conducted using a 3D printed model head similar in scale to quail and mouse heads. This model head included the same frame for fiducial markers used in the surgeries and two straight ducts with different angles. A CT scan of the model head was obtained, and the coordinates of the four markers and the openings of the two ducts were marked. The model head was then positioned in different orientations, and the coordinates of the markers and duct openings were measured using both the mechanical XYZ positioning system and the optical tracking system. The mean Euclidean error was significantly smaller when using the optical tracking system compared to the mechanical system. The errors along the z-axis were greater compared to the x and y-axis, but less so when using optical tracking. The optical tracking system was superior due to its reference frame being fixed to the room, while the mechanical system's reference frame moves with the arm. This means the arm's angle in the mechanical system must remain constant during measurements of the fiducial markers. In vivo assessments of the method's ability to target deep brain areas for electrophysiological recordings were conducted in 3 animal models: barn owls, quails, and mice. In barn owls, the NeuroPixels probe was used for recording in the entopallium, a visual area in birds that was chosen because it is easily distinguished in MRI and histological slices. The probe successfully penetrated through the center of the entopallium as planned. In quails, the method was used to implant NeuroPixels in the hippocampal formation and the dorsomedialis anterior thalamic nucleus. The dye trace verified the positioning of the probe in the superficial hippocampal formation and the dorsomedialis anterior nucleus of the thalamus. In a mouse, a NeuroPixels dummy probe was implanted connecting a superficial target in the retrosplenial cortex with a deep target in the ventral tegmental nucleus. The dye trace appeared in the correct brain areas. These experiments demonstrated the feasibility of the method in targeting multiple brain regions in these animals. The traditional method for stereotaxic surgery relies on fixing the head in a standard position relative to a brain atlas. This method was developed in the early 19th century. However, modern human brain surgery has incorporated advanced techniques like MRI imaging, fiducial markers, 3D tracking, and computerized tools. The method presented here draws inspiration from these techniques in human brain surgery. The primary advantage of the new method is that it allows researchers to conduct brain surgery in animals for which there are no established stereotaxic tools. The method does require a CT scan of the animal and the attachment of fiducial markers, making it more complex than traditional stereotaxic surgery. However, it is especially useful for research in atypical animals such as barn owls. Additionally, this new method is useful for research on animal species that have not been previously studied, because it facilitates precise placement of electrodes and cannulas in the brain without requiring extensive anatomical studies. The brain targets are marked in the MRI image, which can then be reported, equivalent to reporting atlas coordinates. Additionally, similar to traditional methods, a post-mortem reconstruction of the electrode track is necessary. While a 3D optical tracking device is not essential, it provides increased flexibility in probe placement. The optical system performed better than the mechanical system. The authors believe that the superior performance stems from a fundamental difference between the systems. The optical tracking system has a reference frame fixed to the room's coordinates, whereas the mechanical system's reference frame moves with the arm. The method presented here enables precise targeting of multiple brain regions in animals without established stereotaxic tools. While it does not surpass the surgical accuracy of modern stereotaxic devices, it is a useful alternative. Success is determined by visual inspection of histological brain slices. Quantifying the positioning accuracy of electrodes inside the brain is challenging. Most assessments of stereotaxic accuracy rely on visual approximation of the target location and electrode tip position. The method offers a more individualized approach to brain surgery, as opposed to relying on an average brain atlas. The method's cost depends on the positioning and tracking systems used. This method provides a basis for future development of accurate, flexible, and cost-effective alternatives to standard stereotaxic methods. The method can be further enhanced by using in vivo MRI and CT scans for each animal, which could significantly improve accuracy. In summary, this study demonstrates a novel method for performing small animal brain surgery that does not require a pre-existing brain atlas or standard stereotaxic equipment. This is achieved through the use of fiducial markers, CT and MRI imaging, and a real-time tracking system. This method allows for a more flexible and individualized approach to brain surgery and is especially useful for research in atypical animals. This approach is not intended to replace traditional methods, but rather to serve as a complement or alternative to those methods. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.